In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to JAX Tech Talk with the technical information scientists of the Jackson Laboratory.
Welcome to the JAX Tech Talk with the technical information scientists of the Jackson Laboratory. My name is Neeraj Tiwari, and my role as a technical information scientist is to serve the research community with education and practical solutions. JAX Tech Talk is a 15-minute chat that may be of interest to anyone in preclinical research and drug development. So for today's show, we are going to talk about considerations for using Cre-Lox to generate complex new models. For anyone watching live, use the Q&A section to ask your questions. We'll try to answer those live. Today, I have my colleague Dolores Garcia Arocena on the show. Dolores, could you please introduce yourself to our audience?
Hi, everybody. My name is Dolores Garcia Arocena. I have a PhD in cell molecular biology, and my specialty is in neurobiology, actually. I'm currently a scientific engagement manager at JAX. Collectively, I can say that Neeraj and I have helped hundreds of researchers troubleshoot issues with Cre-Lox mouse models.
Thank you, Dolores. It's great to have you on the show today. So for all of you who are new today, we have done many of these tech talks focused on Cre-Lox. I would highly encourage all of you to revisit these talks when possible. So the simplest Cre-Lox system to generate a knockout mouse model has two components that need to be genetically engineered in the murine genome: a target sequence surrounded by LOXP sites and a Cre recombinase gene controlled by a specific promoter. Typically, researchers first generate the two necessary strains and then breed them together in order to generate the desired knockout mice. There are situations where we have to breed multiple flocks and Cre strains to generate a complex mouse model for your research goals. Some of the important considerations for breeding these complex Cre-Lox models. First thing is to consider that there are over 125 Cre recombinase expressing strains that are available from the JAX mice repository. With so many choices, how can you decide which Cre strain will generate the right knockout model for you? The choice largely depends on when and where you want to target the gene knocked out and the genetic background that your mouse model must be for your data to be meaningful. Second thing to consider is to identify which chromosomes harbor the mutant genes. You can find this information on the Mouse Genome Informatics page. If mutant genes are on the same chromosomes, it may be very difficult and sometimes impossible to generate the complex mutant mice you want. Third thing to consider is to ensure that your genotyping assays are specific. Primers for wild-type flocks and Cre can genotype wild-type alleles, flocks, and Cre alleles. For example, if you run the primers for flocks gene A against the gene A and B flocks mice, it should only show bands in the gene A flocks mice and not gene B flocks mice. And vice versa, if you run the primers for flocks gene B against the gene A and B flocks mice. Fourth thing to consider is many strains expressing Cre have phenotypes that were not obvious or characterized when the mice were created. One such is off-target Cre expression. Off-target Cre expression occurs when Cre is expressed in tissues or organs not directly dictated by the promoter used to drive Cre expression. So if this happens with your Cre strain and you are unaware of it, then your flox genes may be deleted in unexpected tissues. This can present you with potential problems, mainly with interpreting your data inaccurately. Therefore, make sure that you are well informed about the expression pattern of your Cre strains. This starts with reviewing the published literature to see which tissue and organs have been examined for Cre expression. When investigators generate new Cre strains, they of course look for Cre expression in the tissues where the promoter should drive in. but they often don't verify the absence of Cre expression in the other non-target tissues. If Cre expression information is not available for your strain, then you should cross your Cre strain to one of the numerous Cre reporter strains that express a visible marker, such as beta-galactosidase, alkaline phosphatase, luciferase, GFP, and other fluorescent proteins in cells where Cre is expressed. Only a single cross is needed to generate mice that carry both the Cre transgene and the Cre reporter, which is all that is required to check that Cre is expressed in just the cells you want. In the next half of this tech talk, we'll share a case study involving a multi-step breeding scheme of Cre-Lox mice to generate a complex KPC model of pancreatic cancer. Over to Dolores.
Thank you so much, Neeraj. Thank you for those considerations. There's more considerations even that we will share as resources in a thank you email that you will receive. But in this next part, I wanted to share a case study, a real case that we came into the Jackson laboratory to highlight the resources that you may consider using the early stages of future models that you may be planning to create. To make a long story short, a group of scientists spent over 2 years trying to recreate a Cre-Lox mouse model of pancreatic cancer, also known as the KPC model featured here in the slide from the Olive Lab. So as you can see in the slide, the KPC model has 3 different mutations. It has oncogenic KRAS mutation, P53 mutation, and PDX1 Cre, that what it does is express Cre recombinase in the pancreas. So if you can see there, the K stands for the KRAS mutation listed there. KRAS is in chromosome 6. The P starts for P53. And there you show it's similar to a knockout effect when that expression gets expressed. That is in chromosome 11. And the transgenic PDX Cre, it got inserted in chromosome 8. So from this standpoint of one of the considerations that Neeraj said, the 3 mutations are in 3 different chromosomes, so getting these 3 in one model shouldn't be an issue, and it's not. What's important to notice there is that each mutation on KRAS and P53, it's impeded to express because of a LOX stop codon LOX sequence. And so only when Cre is in the model, that stop codon will be deleted, and then the malignant mutation will be expressed. So that's what the KPC model is about. And this was published by Hingorani et al. From the Olive Lab in 2005. And as you can see, the mutations are in the 3 different chromosomes, so that shouldn't be an issue. The original model was done in mixed genetic background. The Jackson laboratory also has the congenic versions of that. So in the next slide, number 4, we are going to see the breeding strategy to generate the KPC model. In the paper was published much later by Lee et al. In 2017. They have a nice figure there. And just keep in mind that they used the congenic strains. The 3 of them were on Black 6 background, as opposed to the original lab. So the first step is just to breed KRAS mutant with a P53 mutant, and then you generate a double head. For that, you need, of course, to genotype the litter to make sure that they have inherited the 2 mutations. And then you get the double mutant, and you breed it with Cre. And in the end, you're going to get the mouse model by genotyping the litter. You're going to select those mice that are positive for Cre, that are positive for the TP53 and KRAS. So according to the Olive Lab, these mice generate pancreatic cancer, almost, he claims 100%, other researchers get less penetrance than that, but that is basically what the Olive Lab KPC model is about. So in the next slide, I wanted to point you to the MGI database to appreciate the curated information that you may find on Cre-Lox models, using the Olive KPC model as an example. The MGI database, it's such a wealth of information, and I only have time to touch on a few things, but the main thing that I wanted to tell you is that you can go in there and find out the combination of the 3 specific mutations from the Olive Lab and look at what does the literature say about what's the phenotype of that particular model. And so I'm not going to read it out loud, but you know that is information that is out there and it's interesting. But on the highlighted section of the slide, you'll see that there's so many more mutants of P53, so many mutants of KRAS. So in the next slide, I just wanted to caution you that to be really careful to when you are trying to basically replicate a model that has been published, to make sure that you are picking the correct one, right? In this example, I looked it up. There's 7 PDX1 Cre strains that I know of. There may be more. There's 58 KRAS mutants. There may be more. And over 211 P53 strains. So be really careful when you look into this, specifically when you're trying to replicate the phenotype of a published Cre-Lox strain, whether it is this model or any other model that you may want to see. So on the next slide, I just wanted to share, for example, I went to the Jackson laboratory and typed KPC pancreatic, and I only found 2 different strains, meaning we don't have the Olive Labs model already made. And as you can see, neither of the models that show up here are the KPC model. When you have time, you can look at the recording in more detail, and you'll see that the nomenclature is similar, but it's just not quite the same. So on the next slide, I wanted to pull in a poll question so you can all participate. And the poll question reads as follows. Let me see. A group of researchers, which is the real case that I was talking to you about it, generated a KPC model, but to their surprise, tumors were growing everywhere. So that's when they call us. What may be going on? And so if you could please select all of the choices that may apply. One is they forgot to administer tamoxifen. The other one, they are using a whole body Cre strain. Another one is they are using a constitutive KRAS mutant. Another one is they are using a constitutive P53 mutant. And the last one is the dose of tamoxifen is too high. So I'll give you a couple more seconds here to think about which one of these or which of these choices may explain that the KPC model that they put together after 2+ years of breeding and genotyping, why is it that tumors are showing everywhere in the body, not only in the pancreas? So let's close the poll and see the results. Good job. A lot of you said that they may be using a whole body Cre. That was not the case though. The tamoxifen answers really in this case do not apply, because if you were paying attention, the PDX1 Cre that the olive lab used was not tamoxifen inducible. But I tricked you, perhaps I tricked a few of you, because I did show you that we offer a tamoxifen inducible KPC model, but it's not the olive lab one that this group was trying to recreate. So if we can close that poll and move on to the next slide, I tell you what really happened with these researchers, and I felt so bad for them. What happened was, if you look more carefully at the Olive Lab KPC, they made the mistake in selecting which P53 they bought. The Olive Lab was TM2, which stands for Targeted Mutation 2, which is the one that has the stop flux cassette there, preventing the P53 to express until Cre is in the tissue. But they messed up and they bought from the Jackson laboratory, the one that said TM 2.1 instead of 2. And that one had already the stop flux cassette already deleted in every tissue. So that means that for the P53 mutation to express, it did not need Cre in any tissue. It was just expressing. And so that was the explanation to why this lab started seeing in their offspring tumors everywhere in the body. So if we move on to the next slide, I wanted to show you another application of the MGI that really illustrates what we're going to see. And again, I'm opening here at the MGI for PDX-Cre1. So I was wondering how many other KPC models were made using the exact Cre strain that the Olive Lab did. So I was expecting to see 2 or 3 models, but instead there's 40 plus. And the one that has check mark there is the exact Olive Lab phenotype right there. And in the table below, you can see highlighted also all the phenotypes that the KPC model shows down that column. However, the other versions of KPC using slightly different mutations are different. So my message here is that please pay very close attention when you're trying to recreate a model from the literature, whether it's this one or any other one, to look very well in the resources that we have. You can call us, we can help you, so you can get the model correct from the beginning. So that was the main thing that I wanted to show you. We don't have too much time to delve into the details on how to use MGI as a tool for Cre-Lox model, but I hope that this example sheds some light when we are comparing published models side by side. And also, I hope that you get better prepared when you're planning to reproduce other published Cre-Lox models in your lab.
So, Dolores, I think we can take some questions asked by our viewers today.
Sure.
So the first question I see here is, when we are crossing multi-Cre-Lox strain, if they are on different genetic backgrounds, which mice I should use as controls?
Right. That is a common question, and not every strain is congenic. Sometimes when you start a new flux, it's very typical that it's not congenic. At least the old models were like that. So what you can do is do the breeding schemes in a way that you can get wild types. Well, then you can call it wild types, is non-mutant mice from the same colony. So then you can ensure that the genetic background is as close as possible to that. That would be one way to go about it. Make sure that your breeding scheme allows for some mice that can be used as controls as well.
And the second question we have is why it is recommended that I breed Cre out of my cross.
I'm sorry, can you say that again?
So why it is recommended that I breed Cree out of my cross? So once
Oh,
the
good
cross
question.
is,
Yes.
yep.
Yeah. Yes. So it depends on the situation. It depends on the situation. In some situations, if it's an inducible Cree, you probably want it still there. But if it's a knockout, let's say it's a whole body knockout, or even well, let's stick with that, a whole body knockout. You want to make sure that, you know, at the end of the day, Neeraj talked about one consideration is the expression of Cree may have its own phenotype, right? So just Cree positive mice with no flux and no knockout may have their own phenotype. So you want to make sure to, if you don't need the Cree anymore because it already deleted the regions that are needed and that went germline, let's say, and so everybody, I mean, every tissue in the model has already knockout in this example, then you really don't need the Cree anymore. So you want to breed it out, meaning in the offspring, you select those offspring that did not inherit Cree, just to clean the model up and not confuse it potentially with off-target effects of Cree. So I'm not sure, Neeraj, if you have another take on that, if you want to add to that.
No, I think you covered it all. We have a similar question on the same lines. Should the Cree be homozygous in the cross transgenic mice?
Right. That's a question that we get a lot. And the vast majority of the Cre-Lox models, the genotype of Cree is heterozygous if it's a targeted mutation, or what we call hemizygous, which is similar if it's a transgenic. You don't need two copies to do the work, so to speak. Heterozygous or hemizygous will typically do the job. And to add to that, in certain strains, like we talked about the off-target effects of Cree, homozygous Cree mice are not even viable in some cases. And in other cases, they are not fertile. So it's not convenient from the standpoint of maintaining the colony to have it Cree. I know that from the point of view of you avoid genotyping if everything is homozygous. That's great because then all the offspring will be homozygous, but you have to have all those other considerations as well. And feel free to reach out to us, micetech@jax.org, if you have any particular questions that you need help from us, we can figure it out.
So that's all the time we have for this week. Thank you for joining us. Follow JAX Tech Talk on LinkedIn so you're notified anytime we have a new episode, or subscribe to our playlist on YouTube and catch all previous episodes. As Dolores mentioned earlier, you'll be receiving some useful resources that will be helpful with the Cre-Lox breeding in the thank you email with the video of today's talk. This is Neeraj and Dolores saying stay healthy, stay safe, and stay excited about your research.
Bye, guys.
Bye-bye.